serta syukur kehadiran Tuhan Yang Maha Esa, dikarenakan ridho dan karunia-Nya kita dapat berkumpul hari ini. Yang saya hormati Kepala Organisasi Riset Arkeometri, Pak Dr. Heri Yogaswara yang sudah bergabung via Zoom. Yang saya hormati Profesor Hari Widianto yang menyempatkan hadir pada hari ini. Yang saya hormati Kepala Pusat Riset Arkeometri, Dr. Sofwan Nurwidi. Tidak lupa pula saya ucapkan selamat datang kepada para kepala pusat riset, peneliti senior, kolega di lingkungan BRIN, Dinas Kebudayaan, Balai Pelestarian, dan para sivitas akademik yang hadir secara langsung maupun secara daring di kawasan Sains Baikuni, Bebarsari, Yogyakarta. Selamat datang kembali di kegiatan sharing session dan workshop yang diadakan oleh Pusat Riset Arkeometri. Kegiatan kita kali ini berjudul Using Archaeobotany to Uncover Ancient Behavior and Paleoenvironment, yang akan dibawakan oleh narasumber kita, Dr. Anna Florin. Thank you for coming, and it's a great pleasure to have you here this morning. Sebelum kita mulai kegiatan workshop ini, izinkan saya untuk membacakan sedikit rangkaian kegiatan. Acara ini akan dibuka oleh Kepala Organisasi Riset Arkeometri, Pak Dr. Heri Yogaswara, kemudian akan dilanjutkan pembahasan materi arkeobotani oleh narasumber. Setelah itu kita akan ada coffee break sedikit. Setelah coffee break, setelah itu kita akan mulai berdiskusi. There will be discussion after coffee breaks. So, and then ada break siang. Setelah itu kita ada workshop praktikum sedikit nanti dipandu oleh Dr. Anna Florin. Kita ada floating, ada beberapa metode yang digunakan untuk pengambilan data untuk arkeobotani kayak flotation dan beberapa lain yang lain. Kemudian akhirnya akan ditutup oleh Kepala Pusat Riset Arkeometri, Dr. Sofwan Nurwidi. Untuk mempersingkat waktu, saya persilakan kepada Kepala Organisasi Riset Arkeologi Bahasa dan Sastra, Bapak Dr. Heri Yogaswara, untuk membuka kegiatan sharing session kita kali ini. Mangga Pak Heri.
Oke. Baik, terima kasih kepada Pak Anton. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Shalom, Om Swastiastu, Namo Buddhaya. Salam kebajikan dan rahayu untuk kita semua. Yang saya hormati Pak Sofwan Nurwidi sebagai Kepala Pusat Riset Arkeometri. Yang saya hormati para senior di lingkungan arkeometri dan juga di lingkungan arkeologi lainnya. And also very good morning for Dr. Anna Florin from Australian National University. And also extend regard from Dr. Ari Kim, from the ethnobotany of the Department of Biology. I talked with him this morning, and he hope one day can work together with you also with the new facility in BRIN. I'm so sorry if I speak in bahasa Indonesia for this moment. Oke, sekali lagi saya ingin berterima kasih kepada Pusat Riset Arkeometri yang menyelenggarakan kegiatan ini, yang kegiatan yang saya kira menjadi sangat penting, yaitu bagaimana menggunakan archaeobotany untuk uncover Asian behavior and paleoenvironment. Jadi saya kira ini satu metode yang memang akan kita tajamkan, karena kita akan melakukan yang kita sebut kegiatan riset platform kolaborasi, RIM ekskavasi, di Bumi Ayu khususnya, yang pasti metode seperti ini menjadi sangat penting begitu, Dan juga saya kira di arkeometri ini metode yang memang sepertinya bagi yang peminat arkeobotani wajib hukumnya, untuk memahami metode ini begitu, Kebetulan saya juga kemarin ikut dengan Pak Anton, dengan Pak Thomas juga di Flores, Jadi belajar sedikit juga, bagaimana metode arkeobotani ini, Saya kira ini sangat penting, di dalam riset kita. Yang kedua, saya kira kegiatan workshop ini beruntung, workshopnya luring begitu, jadi bisa dimanfaatkan, dan juga ada juga kegiatan daringnya sehingga orang lain bisa mengikuti begitu ya. Dan ini juga saya kira bagian dari penguatan kapasitas maupun kegiatan yang ada di satuan kerja ya. Saya memang memberi sedikit insentif bagi para kapus untuk bisa membuat kegiatan-kegiatan yang sifatnya kegiatan bersama ya. Kegiatan bersama yang sedapat mungkin bisa juga luring ya, karena kita juga menyadari for human being kita tidak hanya sekedar Zoom ya, tetapi human touch ya, harus bisa saling menyentuh, bisa saling menatap, bisa saling berkomunikasi. Saya kira ini menjadi sangat penting untuk soliditas sebuah pusat riset begitu ya. Dan juga saya harap kegiatan ini tidak berhenti di workshop ya, tetapi bisa ada kerjasama dengan Dr. Stephanie Anna Florin ya. I hope this webinar is not only for webinar, but we will continue with research collaboration with Dr. Stephanie Anna Florin ya. And I think our relationship with Australian National University is quite good ya, especially related with the archaeological research ya. Jadi saya harap ini bisa dimanfaatkan untuk kerjasama selanjutnya. Saya kira itu yang bisa saya sampaikan. Sekali lagi selamat workshop ya, dan mohon gunakan waktu ini sebaik mungkin untuk bisa menyerap ilmu yang dari Dr. Stephanie ya, yang saya kira sangat penting. Apalagi ada berbagai fasilitas laboratorium yang relatif baru yang di BRIN yang mudah-mudahan bisa dimanfaatkan oleh teman-teman ke depannya. Oleh karena itu, dengan memohon petunjuk kepada Tuhan Yang Maha Kuasa, webinar, sharing session, workshop berjudul Using Archaeobotany to Uncover Ancient Behavior and Paleoenvironment pada hari Selasa tanggal 6 Agustus 2024 secara resmi saya buka. Wassalamualaikum warahmatullahi wabarakatuh. Om Santi Santi Om dan Rahayu. Again, thank you for Dr. Stephanie Anna Florin. Terima kasih.
Terima kasih kepada Pak Dr. Heri Suara. Sebelum kita melanjutkan ke narasumber kita, izinkan saya membacakan sedikit biografi beliau. Dr. Anna Florin currently is a lecturer in archaeological science in the School of Archaeology and Anthropology at the Australian National University. Previously, she was a researcher's fellow at St. John's College and the Macdonald Institute for Archaeology Research, University of Cambridge, and also a colleague of mine also at the ARC Center for Excellence for Australia Biodiversity and Heritage, University of Wollongong. She obtained her doctorate degree from University of Queensland in 2021. So I hope that's right? So Anna research is focused on developing long-term perspective on the role of plant foods and their processing and management in world economy. As an archaeobotanist, she studied plant microfossils, wool scraps from ancient fireplace to understand diet of people in the past. She also specialized in analyzing of roots and tuberous vegetables and other tropical plants and works closely with traditional owners and local communities to understand how different plant processing practice, such as cooking and grinding, seen using in archaeological perspective. And her research in Australia, New Guinea, and island Southeast Asia has implication for key topic in global archaeology. This includes the role of plant foods in early human migration outside Africa, ongoing cultural adaptation to climate change, a new perspective on plant and landscape management practices in hunter-gatherer societies. She also actively published her work in reputable journal and presenting her work in global stage, symposium, and seminars. So without further ado, I give you Dr. Anna Florin.
Terima kasih banyak. Thank you so much, Harry and Anton and Pat Thomas and Pat Safwan, and to BRIN for having me here. It's a real honor to be able to talk to you about archaeobotany today. I would just put up. So I hope you don't mind. I've changed the talk title a little bit. Today I want to talk to you about using archaeobotany to understand the human past. And I want to think about it in this part of the world, in islands Southeast Asia, Australia, and New Guinea. Oh, the sound. So why plants? Plants, and this is a direct quote from the FAO Director General, Q Dong-yu, plants are the single most important pillar of human nutrition. Now that might seem like a very extreme statement, but today, globally, plants make up 80% of diets worldwide. This is, of course, supported by agricultural economies and really high intensity farming of things like rice and wheat and corn. But even in societies that are not agricultural, plants still make up really important parts of the diet. So we know from very conservative work in hunter-gatherer societies that in the tropics and subtropics, recent hunter-gatherers have relied on plant foods for 36 to 55% of their calorific intake. And when we think about the idea that plants don't give you as many calories as animal foods, that is a really large amount of plants that's being collected. So human societies in both agricultural and non-agricultural contexts are relying on plants a lot, and they're very important parts of their everyday life and economies. One of the big reasons for this is that plants are, of course, incredibly nutritious. They offer us vitamins and minerals that we may not be getting from other parts of our diet. But also, humans cannot survive on protein alone. We know from early Western explorers in North America that it will take 3 weeks for a person to die if they are eating only lean meats. And this is often called rabbit starvation syndrome. And it is the idea that eating very lean meats, things like wild terrestrial animals and seafoods, is not able to be processed. The protein cannot be processed well by the human body for energy. And if you eat that alone without high-fat foods and high-carbohydrate foods, very quickly humans will not survive on that diet. So in some instances, in very recent societies, especially in extreme environments, places like the Inuits in very, very North America, or in Tibetan societies, we do have people who live on very high amounts of animal foods, but this relies on very high fat, so things like whale blubber in the case of the Inuit, or on dairying processes, so agricultural economies, so using of secondary products of foods that give high fat. But in most societies around the world, plants are incredibly important and are really necessary to human diet. With this said, unfortunately, archaeobotany is not a discipline that is being so heavily focused on in our part of the world, in the Asia Pacific. And this is something that's happening more recently. And it's exciting that it's happening because plants are incredibly important to the ability to get enough nutrients to survive in the world, but they're also important to a whole range of human focuses. So they're important to our social lives. The food we eat is a part of our cultural identity. They're important to maybe religious and different parts of the way we eat with plants. Also in our architecture, and the way we use plants very much shapes our environments. So we know that with more agricultural work, we're really changing landscapes and the way we use plants. So plants are really important to all these ranges of things we do as humans, from things that are very much about how we get enough nutrients to things that are very cultural and very social. So today, I'm going to talk to you a little bit about a few things, but first I'm going to discuss archaeobotany and give you an overview of how we recover and analyze plants from archaeological sites. And I apologize because I know there are some archaeobotanists in the room today, so I apologize if I'm saying things you already know, but I thought I would take it back to basics and talk about that. And then I want to talk about a few big picture questions in this part of the world that I think plants have a lot, that direct evidence from plants from archaeobotany can start to add new ways of thinking about these questions and be very important too. So for those, I'm talking about the big picture questions of early peopling, of more recent Austronesian movements, but also other Holocene movements we now know are going on in island Southeast Asia, and also indigenous developed agricultural practices in Melanesia and island Southeast Asia. So archaeobotany is the archaeologically subdiscipline. It's an archaeometric discipline that's technically concerned with the recovery and analysis of plant remains. It focuses on the study of past human-plant interactions. And this is a whole range of human-plant interactions that we've been talking about, from very, very early times to more recent different types of interactions. It's also called paleoenvironment, meaning the study of the past humans and plants, but not paleobotany, and that is about the evolution of plants. So there's two broad types of archaeobotany that are done by archaeologists. The first is the study of plant macrofossils. Plant macrofossils literally refers to plant fossils that are visible to the human eye. And these include things like seeds and fruits that we so often look at when we're looking at maybe questions about the origins of agriculture in Southwest Asia. But also things like underground storage organs, stem tissue, and also wood charcoal. Alongside this, there is plant microfossils, and these are fossils that are only visible under a microscope. And these include phytoliths, starches, and pollen. So phytoliths, as I am sure a lot of you know in this room, and I know a lot of you work on phytoliths here at BRIN, are sort of the byproducts of plants using both the water in their environment and energy to create themselves, to build themselves. So silica and other minerals also come into the plants with water from the soil and get moved around. And some of those things go into photosynthesis, into making carbohydrates for the plant to build and grow. But other parts of that, including the silica, are not used in that process. And that pieces of silica can be pushed out in the plant in different ways. And they're often in some plants sort of structural supports for the plant to grow. So especially in monocots like rices, they can be used in plants as defense mechanisms against sort of herbivores and things like that. But there is a range of ways that they're found, and they can also at times be identified species. As well as this, we have starch granules, and here you have an image of starch granules in situ inside the Dioscorea yam. So these are the packing tissues, parenchyma, that make up the starchy flavor of that yam and the vascular bundle running through it. And these starch tissues can also be found on tools that are being used to process plants like that yam, and on and in sediment contexts as well. And finally, also we have pollen. And pollen, of course, is part of the reproduction of the plant. It is the male part of the plant that is then put in the female plant to create fruit. And pollen comes in many shapes and sizes, and it's often used in paleoenvironmental research, but can also be used on site, on archaeological sites, to look at the range of plants that are coming into a site. When we're talking about archaeobotany, it's really, really useful to have a range of all of these things when you're doing your archaeological research, because some things are better preserved as plant macrofossils and can be very easily linked to humans because they're charred and we can see them in fireplaces. But some things don't preserve that well that way, and then maybe they're better preserved as phytoliths. Or we're better to see starches on stone tools to understand how tools were used in the past. So it's really lovely to use all of these together to understand plants in the past. So I also just wanted to talk a little bit about the preservation of plants in archaeological sites. And here I'm talking mostly about macrofossils, which is what I specialize in. And I apologize because the talk will be more heavy on macrofossils for that reason. So in most circumstances, we do not expect plants to survive because plants are at the bottom of the food chain. They're eaten by insects, by small animals, that are then eaten by other animals and get removed from that environment and probably put back into the soil and into nutrients and there to grow other plants. So we don't expect them always to preserve. And they do decay very easily. But there are some instances where we do get plants preserving. And these are when plants are naturally persistent. So this includes plants that have minerals within them that make them survive and not decay, usually in the environment. So here we have some beautiful phytoliths at the bottom of a basket from Çatalhöyük in Turkey. And you can see the white, that is the phytoliths that made up that basketry. So the actual sort of rushes that were woven together are now gone. But because the phytoliths are made of silica, which is basically glass, they survive for a very long time. We also have keltis seeds, and these are found in sites all over the world because they themselves have some calcium in their structure. So they're already very easily preserved, and we find them in very ancient sites linked to early hominins, and probably they're being used as fruits at all different time periods. The other reason we might find plants preserving is because they've been transformed into a state that means that they are more likely to not decay. And there's two main ways we see this archaeologically. The first is when plants are charred, and we expect this to happen when they're burnt in a fireplace. Sometimes an archaeological site has been burnt completely and we get whole areas of burnt remains. And that can often allow plants to survive back tens of thousands, maybe even hundreds of thousands of years. The other way that plants are transformed is through mineralization. And here is an artist's interpretation of latrines at Pompeii. And plants being put in with lots of minerals from human feces means that they can preserve and take on those minerals and become mineralized. And so we can get lots of plants preserving in that way as well. The third way that we might have plants preserved is when the environment prevents or slows decay. And there are some environments that are particularly useful for plant preservation. This includes waterlogged environments, very dry environments, and very cold environments. And you have some examples here. This is 300,000-year-old spear from Schöningen in Germany, which is preserved as wood because it's been waterlogged and kept in an anaerobic sediment for such a long period of time. This one is flower garlands from Ramses II's tomb, and because it's very dry, these plants have also been able to preserve in this environment. And the other sort of environment that we find plants preserving in is in dental calculus. And here we're talking very much about plant microfossils that are found in these sort of mineralized crusts on people's teeth. So there is preservation bias in what survives archaeologically. And here I talk mostly about charred assemblages because these are the majority of assemblages that we look at worldwide and that I look at myself. So charred plant macrofossils can preserve over millennia, but they preferentially preserve two types of plants that come into contact with fire. So if you were to eat a piece of fruit and throw it away in the environment, not next to a fireplace, probably never going to preserve. In fact, it's probably not going to show up archaeologically at all. But if you were cooking and you're eating that piece of fruit and you spit it out into a fireplace, then you have a chance that that bit of nutshell from the fruit preserves over time. And also, you're more likely to have things that are coming into contact with fire, so things that are being cooked and processed around fireplaces, preserving for that reason. The other thing we have is that certain plant remains are more robust than others. So here we're talking about things like seeds and fruits, but also nutshell and wood. And we now also think that we find a lot of underground storage organs, but the reason why we're now realizing that these actually do preserve when we thought they didn't preserve so well is because we have gotten better analytically about being able to look for them and recover them from archaeobotanical assemblages, where in the past they were thought to just be pieces that couldn't be identified and weren't played with. So when we're talking about recovery for archaeological plant remains, it's important to remember that they are usually small and mixed up with soil and other artifacts in sites. So you can obviously take radiocarbon samples and you take charcoal, but that won't give you a representative sample of what you're finding when you're excavating. As well as this, plant microfossils are obviously not visible to the human eye, but they're more likely to preserve in quantity, so we don't have to take such big samples for them. And it's really important that we develop specialized sampling techniques to recover them, and we implement these systematically at archaeological sites if we want to know about plants in the past. So one of the biggest ways of recovering plant remains is using flotation. And here I have the design of a flotation museum from, flotation machine that was used in is used very often in Southwest Asia, looking at origins of agriculture type sites. So the idea is you have a context of sediments, and here we might talk about something that is, say, maybe 60 liters or 100% of a context. That is the usual amount. So it's a lot of sediment that you're using to get out the plant remains. You pour the sediment in the top here. There is a sieve that is left here, so some material that is allowing you to capture the heavy artifacts that fall to the bottom. So it's also doing wet sieving essentially for you as you go. Soil then builds up at the bottom of the tank and falls through that sieve. The floating remains are pushed up with water over the top and caught in another sieve here. And I have an example, and I'll tell you, we'll do a little bit of flotation later, but not with the machine assisted. But these are the flotation bags that I use, and they are 250 micron in diameter of sieve size, so very, very small, so we can capture a whole range of plant remains, including very, very small seeds. So flotation machines are really important in environments where there is not a lot of water and you need to recycle water through the machine. But they're also, and in environments where you don't have a lot of people to help you. So they're very equipment heavy, but you don't need as many people. You can do it as one person alone on a flotation machine. In places where we have water to use, and we have people to help us do it, I think the simplest way of doing flotation is bucket flotation. And we'll practice that today. But essentially, it's the same idea. We are lining up several buckets in a row and putting in the soil from our context, adding water to each of those, and swirling it around and allowing the material to float to the top. You're then pouring material into a flotation bag. So here is actually Malantola holding a flotation bag with a bucket where we've been working in Malolo. And all of these charred remains come in here, and then you wet sieve the material that doesn't float. So those are for charred sites. If we have a waterlogged site, then the recovery is a little bit different. Waterlogged sites preserve plants incredibly well, And so there's very high concentrations of plant remains. So it's okay to take much smaller samples when we're working on a waterlogged site. And so often plant remains will be collected like this, or as column or block samples, and taken back to the laboratory. The only thing that's really important when it comes to a waterlogged site is you keep the material moist, because that is how it was preserved in water. And if you dry it out, it will disintegrate. Then to look at waterlogged material, we work in the lab using just geological sieves and slowly process out the material, disaggregating it. So it's a very simple process of wet sieving that's used for waterlogged material. I also wanted to talk a little bit about how we collect plant microfossils. So it's important to do this systematically as well. A much smaller amount of material is needed again for plant microfossils because they are very prevalent on sites. So often we talk about collecting a heaped spoon of soil, and that gives us enough sediment to look at things like phytoliths and starches and pollen. We can take those from every context we work on at a site and connect it to the flotation that we're doing on those contexts. We link those two samples up and we can start to build really interesting patterns of what's going on at the site. We can also obviously sample microfossils from artifacts. And one of the main places that that's done is using sampling on grinding stones or in other lithic artifacts to understand how those how those stone tools may have been used in the past. The third way we could do this is on dental calculus, which I've talked about as a great preservational environment for plant microfossils. So we have sampling on site, but we can also do sampling off site and comparing things that are produced and brought in by humans to a broader environmental signature. And it's very useful to understand how environments change with changing human behavior, to have lake and other environmental cores that are off-site to understand these sort of concurring changes in the past. So now I'm coming to how we analyze these things. And first I'm talking about plant macrofossils and charred plant remains specifically. So very much like we were doing with the wet sieving, I sieve all my samples so that when I look at them under a microscope, I don't have to change the focus up and down and up and down. And it makes sure that I see the very big things and then the next and the next and next. And it means that I don't miss small things. Broadly, using light microscopy, the first thing I do is sort plants into different categories of plant remains, things like seeds and fruits, chaff, so the things that sit around seeds, so maybe like the glooms of rice, nutshell or fruit shell, which we call endocarp, vegetative parenchyma, which is the material, the soft starchy tissue from underground storage organs, other pieces of stem and those sorts of things, wood, which I do not analyze myself, but it's also a really interesting way of looking at paleoenvironmental changes and fuel use in the past. And also the modern materials, so things that are not charred that we don't actually expect to be part of that archaeological assemblage, little rootlets and things like that that have been growing in the soil. For plant microfossils, processing samples, and I know a lot of you do this, so I won't go into it in too much detail. We often will chemically process soil samples or look for sort of visible samples on residues under a microscope. And here you have Richard Fulliger looking at some material, I think from Muncher Baby, to look for these use wear traces, so places where it looks like those stone tools have been used and connecting it to the residue that's on it. So then doing a spot sample of what he's looking at. And then we observe microfossils across slides systematically, and we use different types of light, so things like polarized light to pick up starches to see the different types of microfossils. Now, identification is done in two ways. We can talk about identification using morphology, and this is an example of that here, using the size and shape of a plant to understand it. And the other way we can talk about it is anatomy, and I'll show you that in a second. It's incredibly important to use good modern reference collections to identify what we have in the past. And it's really, really hard to take our identifications beyond just things like nutshell or stem tissue from a monocotyledonous plant without these good reference collections. So you can see here, this is rice, and it's got some very specific grooves that we can then see in the archaeological rice. And so that's a morphological identification. We can also identify plant macrofossil material anatomically. So this is using the cell structure of the plant to identify what it is. And this is really important for looking at material like underground storage organs or stem tissue that we're not expecting to get the whole piece of the plant. We're only getting little chunks. So we don't have an idea of what the shape is. We don't, it breaks up a lot. It's not a little seed that's relatively solid. Also, I use this for identifying very fragmented nutshell. So here you can see this is Nymphea violaceae or Wyok that I collected in Northern Australia. And we have here a histological thin section that was made from this, which gives us an idea of what that plant anatomy of the stem tissue from that plant looks like. And we've got two beautiful vascular bundles, which are the part of the plant that brings in the water and the sugars through the plants, through the phloem and xylem. And they are sort of in a mesh of aerenchyma, which is a type of tissue that has air in it and is often found in water plants to allow them to move air around them. And also we have an air duct in the middle between them. This is an archaeological piece from Baby from about 3,000 years ago. And we can see these vascular bundles on either side of the air ducts. And it's changed a lot, though. It doesn't look the same. And the reason for that is that this archaeological piece was dried out before it was charred, as compared to this one that I put straight into alcohol so that I could take it to the lab and do the thin section. And so it's still very soft and round. And this one's shrunk inwards. So we have to understand the processes that happen. Both we have to understand the anatomy, but also how they change with things like drying or charring. The other method that's used a lot in identification of plant remains is quantitative measurements. And here is an example of looking at the differences between domestic and wild wheat grains. And we're using length and breadth here to understand changes over time. So there's very systematic ways of doing this, and they can be used for identification, but also for understanding how plants have changed in the past. And these sorts of quantitative analyses are also being used in plant and microfossil analysis. And here we have it for starch grains from a site in Guinea. But I want to just put a little warning there, because starches are a very tricky plant, and especially in a tropical environment, there are so many different plants that they could come from. And we know that the starch grains themselves are not connected to the reproductive part of the plant. And so they don't always vary in a way that is taxonomically sort of, the same family might have very different starches in it, and the same as another family over here. So we have to be very careful to make sure we have a good enough reference collection to make identifications on starches. But we can start to use some of these really interesting quantitative measures to do this. The other important thing when it comes to identification is that we start to make sure that when we're publishing, we're using standardized criteria that other people can look at and can experiment with and can argue with. So it's very important, and especially working in an area where we are really building archaeobotany at the moment, that we say how we identified something, what were the features, and what was our reference collection that we used to come to that conclusion. So that someone else can come along and say, hey, I'm not sure, I actually have some more reference material and it could be this as well. And that's how we get better as a scientific discipline. So obviously, the study of archaeobotany really started in Southwest Asia and in Europe. partially on sites like this, where these big tell sites, where you have layers of the site that have been burnt because of disruptive events that are happening at certain points, probably people coming in and burning these small sort of village sites. And these were these early sites where they're finding really big storage pits full of burnt grains. And we have people like botanists like Hans Helbeck, who really started this discipline and started how we do this. And it's a really, really long history in this part of the world. And in this part of the world, it is very normal to do flotation and to systematically look for archaeobotanical remains. And because of this, people looking at this material are starting to be able to use like sort of big data and look at a whole heap of sites across Southwest Asia to answer the sorts of questions they were interested in. So we now know that, for instance, domestication isn't a quick linear process in the way that we once thought it was in this part of the world. It's a really slow process, and different traits come in at different times and are selected for. We can also do things like look at the types of fields people are using by their weed seeds that are coming out of sites and say, wow, they were cultivating, these plants were coming from a cultivated field, these were just coming from a wild stand. So there's a really long history of this, and we need to build these sorts of things to be able to answer questions in the same way in this part of the world. But I think we have that ability, and we have some really interesting questions that can be answered using archaeobotany. So the big questions I wanted to talk to you about are the early peopling of this part of the world, which I know a lot of you people in this room work on. Austronesian movements and these very complex spheres of interaction that we think are now happening in the Holocene in island Southeast Asia. And also to start to think about how we can work on indigenous developed agricultural practices from this part of the world, because it's not all coming in. There's a big sphere of interaction that's happening with plants and that is slowly developing the domestic plants that are used today. So here we have a map of, I guess, this is, I'm sure lots of you are already familiar with this, but obviously movement from the mainland Southeast Asia, or Sunda, so it would have been the Pleistocene, through Wallacea, island Southeast Asia, into Sahul, which is Australia and New Guinea joined up. And I have the earliest sites from 45,000 years and older in blue, the modern human sites that we know of, although obviously there are some difficulties now that we know that Denisovans were in this part of the world as well. And the older lower Paleolithic sites in red. And I'm sure this isn't a comprehensive list of sites, but I just pulled this together quickly. The really interesting thing about hominins and then humans moving into this part of the world is they are suddenly dealing with very, very different environments to what they had before. You know, we're moving through a whole heap of lines that biogeographers have put in this area where there's big changes, especially in faunal turnover from Asia to Australia and New Guinea. Big, big changes, and people are having to learn how to move into these environments. But also, they're obviously having to do water crossings to get there. So it's a really interesting question of how people did this, and it's a really interesting place to think about the idea of what makes us human, why we've been able to successfully move into this area when maybe other hominin species haven't moved as far. So there's been a lot of evidence for a really big part of this being about maritime technology. So modern humans being able to use boats, and we have really good evidence from a lot of these places, especially for the use of marine resources. So things like fish and shellfish very early on. And this is sort of considered to be a really big part of why humans are able to move into this area, is they're able to have these complex maritime technologies that allow them to use environments, especially when we get to this part where some of the islands are more terrestrially depauperate, so they have less land mammals for people to eat. The interesting thing when we think about this is that seafoods unless we're talking about whales and big dugongs and things like that, are very, very, very low in fat. And today, when you think of diets that are heavy on seafoods, they're also heavy on fatty and starchy plant foods. And so probably we need both, but we haven't had as much evidence for the plants, and there's been a real focus on the maritime part of this. So to talk a little bit about this, I wanted to talk about my research at Mudgebaby. Mudgebaby is a site in Northern Australia that has been used by people for probably at least 65,000 years. It's a really important site to the Mira people who still use it today. And it's also a site that is very exciting, not just because it is old, but because we have a whole range of different types of artifacts to look at to start to piece together human behavior at this site. So things like really interesting lithic technologies, ground etch axes, uses of ochre very early on, and also plant remains, which I think is even more exciting. And the other thing that we have here is that we have a site where people have been coming back to over 65,000 years and living through very different environments. So when people first entered this part of the world 65,000 years ago, the site wasn't that close to the sea. It was maybe 200 kilometers away. It was a slightly drier time during MAS4, and then it would have gone through a wet phase and a very dry phase in the last glacial maximum, where sea levels would have been as far as 360 kilometers away from the site. At about 7,500 years ago, and this is work by Kashi Norman, the seawater would have been rising to the point that it actually comes within 1 kilometer. This is the black line of the marine transgression within 1 kilometer of Mudgebaby. So it goes from being 360 kilometers from the site to 1 kilometer away. And that would have changed incredibly the vegetation around the site. In the next 2,000 years, you have a real buildup of mangroves and estuarine environments around the site on those lowlands. And you also have a, and then as sort of the monsoon rainfall starts to bring a lot of fresh water onto that floodplain and the coastline stabilizes around the site, so it's now 70 kilometers away from the site, we get the build-up and the creation of these freshwater wetlands that are really iconic in this area today of Kakadu National Park. And they're incredibly important culturally and economically to the Mira people and other Binging who live in in this part of the world. So I did research on the plant macrofossil remains from this site as my PhD at the University of Queensland. And the methods that I used for this were archaeobotanical analysis. So as excavation occurred, a 1 by 1 meter squared column of the site, all of the sediment from it was floated in the context that it was excavated in. So 100% of 1 meter squared of the site downwards was floated. As well as that, there were 13 hearths that were excavated at site, so 13 fireplaces, and those were all floated as individual features as well. And then using the material that came out of that, I did morphological and anatomical identification using both light microscopy, but also scanning electron microscopy, which is very important for being able to do the anatomical identification. As well as this, I worked on with the Mira traditional owners. So this is May Nengo, who is the senior traditional owner for the site, and her partner, Jacob Janjamer. And they often go out and collect plants and use them today. And they were very gracious in allowing me to do this with them. And we collected a reference collection. And you can see we've collected things that are charred and dried, but also material that I used to thin sections. So I collected those in alcohol and then used histology to thin section them and make really nice anatomical thin sections. As well as collecting plant remains, we also did ethnoarchaeology. We worked on preparing plants and looking at how what sort of processing requirements are required to make them edible. So things like for this ankinchek or cheeky yam, Dioscorea bulbifera, it has to be cooked, peeled, sliced finely, and then leached in running water. So left in running water overnight before it can be eaten the next morning for breakfast. So we did these processes and also collected the products and byproducts along the way. So we have a reference collection that not only allows us to identify plants, but also to identify the products and the byproducts of different human practices of processing in the past. As well as this, I did isotopic analysis on some of the pandanus nutshell that was found at the site to look at past changes in environment, but I'm not going to talk about that heavily today. So what did we find? A whole range of foods were being used at Mudgee Baby, and this includes things that were being used in that sort of very early phase of human occupation, 65 to 53,000 years ago. So anything that is in green was found in that earliest layer when people were using it. So it's got a whole range species or andujme, which is a relative of the mango, and it's absolutely delicious. And you can just pick it from, you can actually shake the tree when it's ripe and they fall to the ground. Very easy, sugary snacks. To things like Polydrupe pandanus, which its nutshells are very, very useful, but it's incredibly hard to open. And I know this because I tried a lot, and I had people I worked with on site could just do it with a metal axe, no problem. I don't have those sorts of muscles. But actually, to do the isotope analysis I did on them, I ended up using a saber-tooth electric saw to cut into them. So people were doing a lot to open those, and especially 65,000 years ago when there was no metal to do that, it would have been a lot of hard work to get into, but they're very, very high fat and give a lot of nutrition. As well as fruits and nuts, we also found a range of underground storage organs, or these sort of roots and tubers. And this includes Dioscorea yam, a variety that isn't poisonous in this example, and some other yam sort of underground storage organs that we weren't able to identify to species because we didn't have the right reference collection. But we could say things like certain ones had anatomical features that suggested that they would have grown in water. So we're able to place where people were going in the environment, even if we couldn't get the species that we were looking for. As well as this, in the more recent past, we get a lot of freshwater wetlands, underground storage organs. So things like the water lily, and also sedge, little sedge tubers at the site. And so also we have a lot of palm materials from the site, and this includes both the top of the palm that can be eaten raw, but also palm from the stem that has to be processed, not to extreme levels of sago, but actually like a little bit similarly, but much easier. You char this, char it, and then remove the inside and pound it with water to remove the starchy material from the very fibrous material inside it. And the final thing we have as well is some seeds at the site, but not that many. This isn't a very seed-heavy economy. We have mostly water lily and lotus seeds from when the freshwater wetlands came to the site. So this tells us some really interesting things. Those plant remains that we're finding in the earliest layers suggest that people are doing interesting processing practices like pounding and grinding. And we have an amazing lithic assemblage that goes with us that also shows us people doing lots of grinding and seed and processing. And there's some really great plant microfossil work from Eddie Hayes, Richard Fuller, and Jude Field that suggests a lot of these were used for processing plants and also for grinding seeds. So that's happening from 65,000 years ago. We also know at the site that we have these drier phases. And for most of the site's use, we have very similar types of plants that are being used at the site. But the difference in what's going on at the site is in how much energy or how many plants are being need to be heavily processed are being used. And in the periods that are drier, we have more plants that require processing, things like the pandanus and the palm. Then, at about 2,000 to 4,000 years ago, when those freshwater wetlands come into existence at the site, we get a whole heap of freshwater wetland plants, as you'd imagine. These very productive environments are being used by people straight away. But what we can tell from the archaeological record is this isn't just a change in the types of plants that people are using, but a change in the time of the year that people come to this site. So for the main period, so here is a seasonal calendar for the Mira people. And it's mostly got 3 big seasons over the wet and then 3 over the dry. For the main period that people have been here using the site, so from about 65 to about 4,000 years ago when those freshwater wetlands come into place, we have people using a range of fruit that suggests to us that they have to be there in Gurung and Bunumale, which is at the very end of the dry and the beginning of the wet season. In that last phase, when we have the freshwater wetlands, it's a real shift, and we can see people coming in and using the wetlands as they dry out in a very different season in Bangarang and Yeke. So at the beginning of the dry, as these wetlands are slowly drying out, people are coming towards them to use them. And we also know that this is probably actually a real seasonal shift because we stop getting those fruit, which we'd expect people to keep using because they're still near the site today. The interesting thing about this is that in Gurung, at the end of the dry, this is a time period where you've had a whole year where in the wet you get this big buildup of plants on this in this area, and then it dries out, and it really dries out. And at the very end of the dry, you have a lot of fuel load that is very dry and ready to have a fire. You then start to have lightning storms on this environment. So if you have that massive fuel load and you also have the lightning storms, suddenly you have big, really dangerous fires. Today, to deal with this, people light very small fires in this environment, and they slowly burn off all the way through the dry, that fuel load that's created in the wet season. And the fact that we have people here at 65,000 years ago living there, and we have the mature fruit trees with them, suggests that we're not getting these massive burning events going through the site, and that maybe people are doing some of these sorts of landscape management practices from very early on to be able to live in this environment. So I think what we see here at Majababy is a very resilient economy in the face of large-scale environmental change. So you have all these big changes, but people keep coming back to the site. And part of how they're doing this is because they have a very broad diet and a very broad plant-based diet, and the processing technologies necessary to use all of those plants, as well as things like seasonal mobility and movement around that landscape, and maybe landscape management through firing. I think we're pausing here for a coffee break.
Kita akan coffee break sebentar, mungkin 15 menit. Ada kopi dan teh di belakang jika Bapak Ibu ingin yang hadir secara langsung di sini jika ingin beristirahat sejenak sambil mungkin mencerna apa yang sudah dijelaskan oleh narasumber kita. Silakan. ada pengumuman sedikit bagi yang hadir di ruangan eh, yang datang hari ini di ruangan 12 hari ini. Kalau belum absen, ada absensi di depan, mohon mengisi agar secara administrasi kita bisa aktif. Terima kasih, mengingatkan kembali. Oke, selamat datang kembali. Mudah-mudahan sudah mulai sugar intake-nya sudah mulai stabil kembali. Jadi bisa fokus lagi untuk melanjutkan presentasi dari Dr. Anna. So, please, thank you.
Uh, so we just finished talking about this work at Majababy and the idea that early people at this site had very complex plant processing technologies and a very broad plant-based diet. And that this allowed them to have a resilient economy, even though there were some big environmental changes that would have happened in this part of the world. So from this, I think that I would like to put forward a hypothesis that diet breadth and the ability to process plant foods to get the nutrients out of them is actually really key to the peopling of these novel environments. And that's alongside the ability to do marine specializations. So these different types of technologies and processing technologies or hunting, fishing technologies is really key to the way that people move through this part of the world. And whilst Majababy is the earliest evidence we have for this, we also have some early evidence from other Pleistocene sites on either side of the Wallacea that also suggest this. So we have the movement of humans up into the highlands of New Guinea 49,000 years ago. And as they're doing this, we know from Andy Fairbairn's work and Summerhays that they're probably using pandanus there as well, but one that's very easy to get into, a monodrupe pandanus. So one, it's much easier to open, but also using these high-fat plants. Maybe as well, they are using yams. But we have to be careful a little bit with that identification, but maybe they're also bringing yams up with them. And we know as well that they're probably, that at least sort of 4,000 years later from being in that environment, they are also using fire to create their environment. So we're also seeing landscape management. And we know from the pollen cores here that by 45,000 years ago, we get these peaks in fire that in this rainforest environment wouldn't happen naturally. So it must be the people burning. And probably burning to get into the rainforest to better hunt animals in there and maybe to create stands of pandanus. But that slow burning really changes that environment and maybe sets the stage for what happens later on in the highlands of New Guinea by creating grasslands in that area. As well as this, at Nia Cave, we have really lovely evidence from probably about 46,000 years ago by work done by Victor Paz and Hugh Barton with Graham Barker, looking at that suggests that people might have been processing some toxic plants there. So we have things like Pangium edgel, which is a type of fruit that requires processing. And the way you process this, and that they think it's being processed at the site because they find pits with Pangium edgel in it, is by burying it for about 40 days. So we can see probably that these early humans moving through this environment were using plants intensively. And while we don't have evidence in Wallacea itself, I'm sure it's the same case, and maybe even more so in an environment where there are less terrestrial animal resources, and we're relying very heavily on very lean shellfish and fishes for our food. This brings me to some work that I'm now doing with Dylan Gaffney and Dal Tanujiro, and also a lot of members of BRIN who are actually out in the field at the moment working in Waigeo. And this is at Mololo Cave on Waigeo Island in the Rajarampa Islands. And this site now has a date for about 55,000 years ago, or maybe older. And this date is on a piece of tree resin that was found in the excavations of the site, and is giving us an infinite age. And the resin is not formed in the way that it would form naturally on a tree, but has been pulled and then stacked by humans. And it might be brought into the back of the site for a range of reasons, but one of them could be to provide light as a really good fire starter in the site. So that's also some really interesting evidence for plant processing very early on by humans moving through this part of the world. And I think this paper is actually coming out in a week's time in Antiquity. When Dylan was first doing this work, he sent me the samples he collected for radiocarbon dating at the site, and I looked through to see if any of the material was not wood, but in fact pieces of plant foods. And amazingly, considering we only looked at about 30 samples, a lot of them were plant foods. And we've got some images here of vegetative parenchyma, so tissue from a monocotyledonous plant. So these are plants like taros, yams, are all monocotyledons. We've got tissue from that as part of the material that we dated at the site. And in fact, that piece at the top there is coming out at 13,000 years ago. So I already knew there was potential for this. And, oh, go back, sorry. I'm sorry. In the last field season, we also did flotation at the site, and I'll talk about it a bit more, but we're starting to look for plant macrofossils, which I know are well preserved at this site. So it'll be very interesting to see that early picture in Waigo Islands. So how does this differ with modern humans maybe to other hominins who are in this region and didn't come as far across the Wallace Line and into Southall? Interestingly, there's a little bit of data that Mike Morley and Pat Thomas as well has worked on from Liangbua. And this is suggesting that whilst there is a lot of evidence for cooking and using fireplaces in the early human record at Liangbua on Flores, in the earlier record, the geomorphology suggests that fires weren't being lit at the site. I'm not suggesting at all that earlier hominins weren't using plants. I think they would have been using plants extensively. Potentially, though, the difference we are seeing is the ability of modern humans and maybe other closer ancestors, people, maybe Neanderthals and Denisovans, and we don't have the data to pull these apart yet, being able to do these complex plant processing procedures, whether that is just being able to light fires and to cook plants in controlled ways, which is really important, for instance, when we're looking at underground roots and tubers, where you don't have starch that is easily digestible until it's cooked. Or, for instance, mechanical grinding, or using heat to open things, all these different types of techniques. Maybe there's a big difference here in what different species could do. And I think it's as interesting to look at plants and how people are using them and direct evidence for that as it is maybe to look at stone tools when we think about the cognitive capacity of humans and our hominin ancestors. Okay, so now I move to another big picture question in this region that I think archaeobotany has a lot to say about, and this is the movement of Austronesians. And I think everyone in this room knows the very orthodox story of this that was put forward by people like Peter Bellwood, which is the idea that we have the spread of this language family with a group of people who have maritime abilities, but are also farmers. And they make rice and they make pottery and they move outwards through this area. And this was once thought to be a very strong evidence for the dispersal of language with farmers. And while there is a lot of evidence for movement of people in this area from who are probably linked to these languages, we're starting to see that there's a lot of complexity in this time period in island Southeast Asia, and a lot of different things going on. And this comes from a range of different evidence. We have a lot of new papers in human genetics, and I'm sure some of you have worked on these, for instance, that suggest that there's not only movements of Austronesian matrilineal groups, but also movements of groups from mainland Southeast Asia, and also west from New Guinea back into island Southeast Asia that are happening probably in the late Pleistocene, early Holocene. And also newest evidence is suggesting that this is actually happening in the same period of time, especially the movement back from New Guinea, as Austronesian people are moving. So we're seeing a much more complex picture. It's just not just one group moving through this area, it's other groups moving around and bringing things with them and different ways of living with them. The animal evidence for these movements as well has become more complicated. A lot of the animals we're talking about and we thought were very much definitely Austronesian movements are also being shown to potentially move from mainland Southeast Asia as well as from Taiwan, but through places like Vietnam and Thailand. And there's also increasing evidence for a really interesting indigenous maritime network in this region in this time period. And this is evidence like things like obsidian, which Kristen Riefmeyer has been showing has been moved from islands, different islands around island Southeast Asia. Stone adzes, which were once thought to be an Austronesian technology. And shell bees and hooks, which suggests we have populations that are interacting with each other and finding ways to symbolize themselves as different from another group. So we have this really interesting thing that's happening probably pre-Austronesian, and the Austronesians are linking into these trade and these maritime technologies that are already happening. And the last part of this is the plant evidence. There is not any evidence for rice across the Wallace line with Austronesians. There is evidence for rice in mainland Southeast Asia, but there's no evidence into the Wallacean region. So this idea that we have Austronesian groups going out with rice, there's not good evidence for it at the moment, but there is also less archaeobotany than we would like. So we cannot always say that absence of evidence is evidence of absence. But at the moment, we don't have evidence for that. What we do have evidence for is a range of plants that are likely being indigenously domesticated in Melanesia, and maybe also in island Southeast Asia. And of course, the best evidence for this comes from Cook Swamp. And this is work by Jack Golson and then Tim Denham, and a range of other people with them, who really showed that you have these managed agricultural landscapes in these highland areas from about 6,000 years ago. And they connected these managed landscapes into at least the growing of one plant, which is the bananas. And they found a whole heap of banana phytoliths in these anthrogenically created landscapes for growing. But there hasn't been so much other archaeobotanical evidence for this. In fact, it's more about the earthworks and these big managed landscapes than about the archaeobotany. So I've been working recently at a site called Machakurutu in East Timor with Sue O'Connor and Chris Clarkson and Susia Sampacaro. And I just wanted to tell you a little bit about the plants we've been finding there. So this is an important site for early human movements. It puts people in East Timor about 40,000 years ago. And then it also has remains from the early Holocene and the late Holocene. So at this site, I didn't do the flotation, but a colleague of mine did go out and do some flotation. And they floated all of the sediments from the Holocene in one square, in square B, up to Spit 30. So we don't have, unfortunately, flotation from the Pleistocene, but we do have some flotation from the Holocene that's allowed me to have a go and see if we can find some of the plant macrofossils and start thinking about, are there things that are being brought in, or are there things that are being indigenously developed there? And to help with this, we've had a master's student at the University of Oxford who's been doing direct radiocarbon dating on the plant remains because the site is a little bit mixed. So we've been having a little bit of trouble telling that story until we're starting to actually get direct dates. So all of the material that was floated came from these two layers. And when I first was working there, I was very excited because I thought we'd have the early Holocene and then the late Holocene, and we could see what changed over time. But unfortunately, I think there's a lot more mixing than we realized. So we're really starting to find that maybe most of the archaeobotanical assemblage is coming from the late Holocene. But it's still telling us some interesting things. So just a little bit of an idea of the types of plants we're finding there. There is a lot of palm being used and preserved at the site. So here we actually have the endosperm of a medium-bodied fruit from a palm. And the endosperm, when I say that, we're not talking about the nutshell, we're not talking about the husk, we're talking about the fatty bit that you'd eat in a coconut. It's not from coconut, it's too small, but I'm going to talk a little bit more about that later because there's a lot of that. We have stem and petiole material from two different types of palms. The one up here, you can see has these beautiful little globular echinate phytoliths in it. And it also has raphid bundles. So bundles of these calcium carbonate crystals that are very sharp and are used by plants to push, I guess, little bits of poison or irritants into herbivores that are trying to eat them. And from that, we know it has to be one of these types of palms, but we haven't gone any further because they don't have reference collection from this area. We also have another type of palm that we're getting stem, petiole, so that is the branch that the leaf comes off, and also the leaf. We also get a lot of bamboo stem in the site, which is not surprising because we know for this area that there has probably been a lot of bamboo material and a lot of tools made from bamboo, but we haven't been able to show it archaeologically as well. So this is bamboo stem. And then we've got a few little bits and pieces of fruits and seeds. So we've got echinocloa seed, which is probably a wild millet from the area. A few of those, some keltis, which we talked about, sort of appears in lots of sites around the world. Pandanus fruit. And also, we're very interested to find these sorts of clumpy bits of food stuff, so a porridgey type material, which had bits of rice and rice husk in it. And the rice husk was very beautiful to look at because you can actually see the phytoliths on the back and the front of it. And these bits, these clumps of rice were stuck in food-like materials. And this came from context B5, which is the late Holocene context. And we were very excited. Did this mean that we were seeing rice being brought in at this time period, about maybe 2,000 years ago, or maybe not? So Sylvia did the radiocarbon. Yeah, okay. Sylvia did the radiocarbon dating on this, and it was all modern. So it's all coming back from the historical period. And so we've got this porridge with rice, but it's probably from very recent times of people using this rock shelter. And this is something we have to be really careful with, especially when we're looking at rock shelter deposits. They can move a lot, so we have to be really aware of the toponymy in the sites themselves. So I said I'd return to this palm endosperm. So as I said, this is this part of the palm fruit, the fatty bit of it. And it is only that part of the palm fruit that's been preserved, and it is the most preserved material in sight. So I think of about 1,000 pieces we looked at, 700 pieces were this palm endosperm. So that is a lot of this fatty bit of the palm fruit to be preserved when it is the bit we least expect to be preserved. If you imagine a palm fruit being charred, we'd expect maybe some husk, definitely the endocarp, the bit that is really hard to get into. We don't really expect this sort of oily palm fat to be preserved. So it got me thinking, why is that happening? And just to let you know, there were 2 pieces of nutshell in them from the very recent spits, and they were from coconut. But this palm fruit is way too small to be coconut. So I wonder if what we're seeing here is potentially a copra style processing. Copra is the dried coconut endosperm used to make coconut oil. So when this is being done, the nut is separated from the husk and then broken in half. It's light in the sun, or if it's rainy on a smoky area, and the actual fatty piece of the nut comes separate from the nutshell. So I'm wondering if potentially what we have is people using not coconut, but a medium palm fruit in a similar way to the way they process coconut for oil. It's an interesting idea. I think that the fact that we have hundreds of pieces of this and only 2 pieces of nutshell from the site is very strange. It's not how you'd expect preservation to happen. So I think there has to be something in the human behavior that's making that. And this is my best explanation for it. And this, we're getting direct dates back to about 3,000 years ago. So at Machu Picchu, we have very mixed contexts. And the really lovely thing about archaeobotany is we can directly date what we're looking at. So even if contexts have issues, we can see it when we date things. So it's a really useful material to work on. The rice foodstuffs themselves are modern, and they are probably people in historic periods using the site, making porridges and adding rice into it. I guess this doesn't mean that we won't find rice across the Wallace Line. This is only one site, but it's definitely not here. And Ian Glover's early work and also Nuno's work in East Timor has suggested that there is probably not rice in this area. There's been a lot of early archaeobotany here. What is interesting is we do have the use and processing of palm fruits from at least 3,000 years ago at this site. And this is not a coconut, it's probably of an indigenous palm fruit to the area, something that's a medium size. And then alongside this, we also have some fruits and seeds that are being used. But most of the assemblage I'd suggest is being used as technology. So things like bamboo and all those different bits of the palm leaf are likely coming in to be used to make things. And we see this all the time today. So I think what's interesting here is we're not getting new things coming in at this point when we get pottery coming into the site. We're mostly seeing indigenous plants to that area. So that brings me to my last big picture idea that I wanted to talk to you about, that I think archaeobotany is incredibly important to this region to start looking at better. And this is the idea that, and this is from Denham 2020, that this whole area is probably a sphere of interaction with a range of plants, including things like sugarcane, banana, yams, and taros that are being domesticated. It's probably not just happening in the highlands of New Guinea. These plants are found across this whole area, and we need to start looking at sites to see if we can start to pull apart what is happening, the practices, how people are using plants over time and how that changes, to be able to start to answer questions about domestication in this part of the world. Of course, these plants are trickier to work on than grasses in Southwest Asia. So we're going to have to develop the analytical techniques to do this. We need to use anatomical identification to identify things like taro and sugarcane. We need to use phytoliths for bananas, and that is already work being done to try and look at different changes in bananas and different varieties. And we need to use these together. But I think the important thing in this part of the world is also to be able to think about the human practices, the behavior of cultivation or of using forests versus gardens versus intensive agriculture, and how we look for that archaeologically. So I wanted to return to the work at Mololo Cave that I've just been doing. And here we are now doing flotation. And we have a full column from about that 55,000 year date, but also including these Holocene and late Pleistocene and Holocene middens at the site that has been floated, as well as some hearths from the site that we've floated, so some fireplaces. And so this archaeobotanical analysis that spans these periods of time is likely to start to be able to, in connection with all the other archaeological evidence, start to tell stories about how plants are used and how that changes with time and with different movements of people into this area. And Mololo Cave also has pottery coming in that in the Holocene period, which includes red slip ware that is linked to Austronesians, but also corded ware and different types of pottery that are linked to other groups. So it's an interesting place to do this. But the really important part of this is not just looking at the archaeology, but creating the reference collection, and a reference collection that is an ethno-archaeological or ethnobotanical reference collection. So it's linked into the way that people process and practice and use plants, so that we can understand those behaviors and how we might look for them archaeologically. So in this last field season, we've been working with some of the local women in Wasamban village. And here we are in Mama Membrasa's garden, and working with Mama Membrasa and Mama Marina, and also with Poppy, who's an ethnobotanist with BRIN. We've been working together to record these traditional processes. And in this season, we were really lucky to work on sagu. And as we recorded how these sagu are processed by these women, we recorded local names for them. We recorded variations in how they might do this, but also we took samples from every step along the process so that we can see the different products and byproducts of sagu processing. So we can look at the hella, and then the hella after it's been processed in water, and then the sagu. And we're hoping that this will form the basis of a reference collection to start looking at these sorts of things in the past. And this reference collection includes reference material for phytoliths and for microfossil remains that will stay with BRIN, and reference material for plant macrofossils that we will process and have 2 copies of this reference collection. So there'll be reference collections that will allow other people to use them here. So we hope that that will help other archaeobotany in this area, and also for Alifa and Adi, who will be working on the phytoliths at the site. So thank you so much for listening to me talk about archaeobotany. I really want to make the conclusion that archaeobotany has the ability to provide direct evidence for these big picture questions in this part of the world. And I think these are questions that we're all really excited about answering, and span long periods of time and lots of different parts of human life in the past. And I think if we do the right work, which requires method development and systematic archaeobotanical recovery, so we require more flotation, but also more collection of microfossils at archaeological sites to be standardized in the way we do work in both island Southeast Asia, in Australia, in the Pacific, we can start to answer these questions. And as well as this, a really, really important part is the building of reference collections, and reference collections that don't just let us identify plants in the past, but working with people today to understand practices of processing, let us identify human behaviors connected to those plants. So the ways that people make things like cooking and grinding and leaching and all these different things. And so it's really important that those are connected in, and we do really interesting work to work on identifications, and to build our ability to look at plants in the past in this region. And I'm really excited because I know that these are the same conclusions that some of you have come to working here, that the archaeobotanists in the archaeometry team have come to, is also that we need more archaeobotany, and we need good reference collections, and we need systematic recovery as part of our excavation. And that's how we're going to start to do really interesting work in this part of the world. Terima kasih. Thank you so much for listening. And thank you to everyone involved in my research.
Thank you very much, Anna. Such a great presentation and very complete information. I think it's just huge, and we might need times to digest everything. But anyway, thank you very much for the presentation. Untuk teman-teman yang hadir di sini, mungkin ada pertanyaan yang hadir di Zoom meeting, di Zoom dan di YouTube juga. Mungkin jika memang ada pertanyaan, silakan langsung ditanyakan. Bisa dalam bahasa Indonesia, nanti bisa ditranslate juga ke Anna. Mungkin ada pertanyaan? Silakan Pak Joni.
Terima kasih. Mohon maaf saya menggunakan bahasa Indonesia karena saya bukan penutur jadi bahasa Inggris. Ini alasan klasik ya. Nona Florin yang baik. Saya menekuni ilmu linguistik historis komparatif. Jadi kalau berbicara makhluk yang namanya Austronesia ini saya tergelitik. Karena bahasa Austronesia ini adalah bahasa yang dihuni hampir separuh makhluk yang ada di dunia ini, dari mulai Madagaskar sampai ke Rapauni, begitu. Dan Indonesia mempunyai posisi yang sangat strategis. Nah, ini alangkah sangat indah jika problematik permasalahan kebahasaan terutama Austronesia yang di Indonesia khususnya ini menjadi posisi strategis, ini belum terjawab mulai dari Aidean Blas sampai juga Peter Bellwood bekerjasama dengan Blas itu sudah berkolaborasi tentang arkeologi dan bahasa, namun belum sekomprehensif karena memang di Indonesia ini sangat beragam, ada Austronesia dan ada non-Austronesia. Yang tadi Nona Florin mengatakan tentang garis Wallace. Garis Wallace ini kan sebenarnya sudah menurut Dian dan juga Blas sudah istilahnya masih menjadi sebuah problematik karena perbedaan antara penutur Indonesia Timur dan Indonesia Barat itu dibedakan oleh tanaman yang berbeda dan binatang yang berbeda. Nah, ini sebenarnya sudah terbantahkan. Apakah ini bisa diperbarui kembali? Dan alangkah indahnya jika persebaran bahasa-bahasa Austronesia, tanaman-tanaman pemilik penutur bahasa Austronesia, dan pertinggal-pertinggal dari arkeologi penutur Austronesia dan juga non-Austronesia ini bisa kita kolaborasikan bersama-sama, penelitian bersama-sama atas nama di Indonesia ini BRIN, lewat mungkin nanti PR-nya Mas Sofan, mohon izin Mas Sofan, ini menjadi sebuah ekspedisi yang berkelanjutan gitu. Kelihatannya ilmu ini kalau di linguistik ini ilmu yang sangat kuno gitu, tidak menarik. Tapi ketika itu dikolaborasikan bareng-bareng, saya kira mungkin akan menjadi sebuah penelitian yang komprehensif dan bisa problematik posisi strategis Austronesia di Indonesia ini akan terjawab lambat laun. Terima kasih. Mungkin membingungkan, saya juga bingung sendiri, tapi ini menjadi Indonesia ini menjadi laboratoriumnya penelitian dari peneliti-peneliti luar negeri. Tapi kita sendiri orang Indonesia kok kelihatannya mati segan hidup tak mau gitu. Seakan-akan ini penelitian yang entah berantah, itu penelitian yang sangat kuno. Tapi ketika saya datang ke sini dengan Nona Florin, seakan-akan mungkin bisa gayung bersambut. Terima kasih.
Terima kasih. Thank you. It's such a big and exciting question, and I think it does need a big collaborative approach to answer it. And I would be very excited to be part of that. But I think, it will also need geneticists and archaeologists and a whole range of archaeologists and linguists to come together and do this. And I think that is a very exciting idea.
Begitu, Pak Joni, ada lagi mungkin tambahan? Boleh.
Hal yang menarik, Dona Florin, ini pertanyaan yang sangat menggelitik juga. Ketika saya penelitian sampai ke Papua, dan juga kemarin dulu beberapa tahun yang lalu itu penelitian di NTT, Indonesia Timur, yang notabene itu adalah penutur non-Austronesia atau disebut juga Melanesia. Tapi mereka mempunyai pertinggal yang notabene saya katakan ini adalah penutur orang-orang non-Austronesia tua atau proto-Austronesia yang dikatakan oleh kalau ahli bahasa ini adalah Melanesia. Jadi walaupun mereka penutur Melanesia non-Austronesia, tapi mereka punya kesamaan-kesamaan dengan orang-orang Austronesia. Ini bagaimana? Terima kasih. Dari sisi ada umbi-umbian, ada betatas, ada semacam itu adalah ciri-ciri orang-orang Austronesia lama. Begitu. Terima kasih.
Terima kasih Pak Joni yang lainnya. Mungkin saya menambahkan sedikit ya, mungkin Anna ya, just add something from Flores and Papua. Dan itu betul Pak Joni ya, untuk bahasa memang berbeda ya. Itu yang untuk di Indonesia Timur, kemudian untuk kayak di Timur mungkin lebih didominasi oleh Dawan ya kalau nggak salah ya. Terus kemudian untuk di satunya yang kedua adalah Manggarai. Tapi memang betul ada beberapa kesamaan dengan apa namanya tadi kalau nggak salah Anna sudah menyebut untuk cultural package ya. Jadi artinya itu adalah paket dalam konteks Austronesia maupun baik Neolitik ataupun Austronesia. Dan salah satunya adalah berkaitan dengan super crop ya. Jadi apa namanya umbi-umbian ya. Ini juga saya sayangkan karena kemarin saya dengan Mas Anton baru bulan kemarin terus saya mengumpulkan umbi-umbian yang masih di hutan betul. Dan itu memang indikasinya dari ini mungkin Austronesia atau memang asli di situ. Dan itu namanya juga UI, sama dengan di sini. Jadi itu kan masih secara toponimi ya, itu masih berkaitan dengan Austronesia. Dan itu betul sekali. Saya juga setuju suatu saat kita bisa melakukan penelitian secara holistik ya, dari segi karena bahasa itu yang menyatukan antar ini ya, antar kelompok, antar etnik maupun antar suku di sana itu. Dan beberapa tanaman memang sama. Bahkan saya contoh ya untuk yang kita katakan nyiur apa? Kelapa ya. Itu saya temukan juga di aborigin yang ada di utara. Yaitu mereka juga nyebutnya nyiur sampai ke mana? Ke mana? New Zealand apa? Maori sama juga ini. Terus kalau suatu saat kita bisa melakukan kajian bersama antara yang data-data arkeologisnya, terus kemudian dari paleobotani dan kemudian juga bahasa, ini suatu hal yang novelty betul dan suatu saat mungkin kita bisa menggandeng beberapa kolega baik di dalam maupun luar negeri. Mungkin terima kasih Pak Joni untuk masukannya ya.
Mungkin ada pertanyaan dari Zoom ya, kalau tidak salah. Yang itu bukan yang sebutin atau di sini sebentar. There is 3 questions. Zoom, I think. The first one is, I'm sorry. The first one is in English. So I watched the program Tanira Cave, Neanderthal behavior from Rob Selecki in one scene that showed that Neanderthal burial that used flower, pollen, and manhir sculptures. However, when I searched with the keywords Sanidar and flower people, it said that it is a modern misconception.
Yeah, so it's a really interesting one. I believe that idea about flower, pollen, and burials, or flowers being laid with Neanderthal burials, first came from Shanidar when people looked at the sediments around the burial and found that there was a lot more pollen than in other parts of the cave. I think it's highly contested. There are many schools of thought when it comes to Neanderthals and their behavioral complexity. And there are some teams who do not think those burials are burials, and some teams who think they are. I'm not very well placed to argue one way or the other, although I believe that Neanderthals were very complex. And as we see more and more evidence of Neanderthal behavior, we're finding that they were using a range of plant foods, especially in coastal sites like Figueira Brava in Portugal, where we see them using a whole range of animals, marine species and plants, and complex processing methods. So I think I have every belief that they were able to do this, and they had the cognitive capacity and had cultural ways of doing things, but I think the evidence is contested.
Is there any evidence flower people in either Indonesia or Australia? Like, is it true that natural food modification started from Goblik Tepe and spread through the world?
Okay, is there evidence? There is no evidence for pollen that I know of with burials in Australia. There is some beautiful evidence at Lake Mungo for burials with ochre. So a similar idea, very, very early humans who clearly had an understanding of death, maybe whether it's about the cultural importance of the people in the present or a feeling about the afterlife and where they want to send that person to. There is this process of bringing ochre from many hundreds of kilometers away into this arid region to then be used in burials. And that's a pretty amazing phenomenon, I think, similar to the idea of laying flowers. The idea, no, I don't think that anything was started at Göbekli Tepe and spread from there. There are many, many interesting plant uses around the world, and most of them happen indigenously. And then we have an amazing globalized world today where things have spread, but it's not like there was one group of people who worked out how to do this. There was a lot of cognitive capacity in early humans and probably Neanderthals and Denisovans and our closest ancestors to do really interesting practices with plants.
Okay. Next question is Nurfianto Gerapanidu. Hang on. It's in bahasa tadi. Research from BRIN, like ecology, ethnobiology, and then medicines, traditional medicines especially, and manuscript research centers has been give some information in terms of ethnobiology and local knowledge for traditional medicines. So is there any part of those research could help the archaeobotany field?
100% it can. These ethno-historical or ethnobotanical documents about how people use plants are incredibly important. And definitely feed the way we understand plant use, because there are so many interesting local ways of doing things that maybe aren't common practices today, and they help us think about them and what could have been happening in the past. I will add to that I do think as archaeobotanists, it's important we do our own ethno-archaeological research, because it's that next level as well. Like, I know that there has been research where I'm working about how people use plants and the plants traditionally, but it's that extra level of being able to connect the way it's being done to a material signature that we could look for archaeologically that really lets us do that extra bit. But definitely they get used together and they're very important, yes.
Okay, the next question is, based on the result of the research, is there any correction to center of origin of original agricultural communities that has been found previously, especially from Southeast Asia region?
Thank you. And this is an interesting question. I have to say that my PhD was very much based in Northern Australia, where we do not have a lot of evidence for domestication, at least at this point. And I'm only now moving into working in island Southeast Asia. And as you can see, the projects at Machukurutu and at Mololo are at different stages of completion, but neither of them are properly organized to that level. And I think we'll need a lot more research like those small pieces that I've been doing, I'm starting to do, before we can make corrections to this. But I do think it's important that we think about these areas as places that could be involved in origins of agriculture.
Thank you. Ada pertanyaan lagi mungkin? Ya, silakan. Sebutkan nama dan dari mana ya?
Sebelumnya mohon maaf saya pakai bahasa Indonesia aja. Perkenalkan saya Sabita, mahasiswa dari UGM. Saya ingin bertanya beberapa pertanyaan. Yang pertama terkait hasil proses flotation, seringkali ditemukan biji dalam kondisi yang tidak hangus. Apakah biji yang tidak hangus ini dapat dianggap sebagai intrusi modern atau tetap merupakan temuan arkeologi namun dari abad yang lebih muda? Dan bagaimana membedakan biji modern dengan biji dari masa lalu tersebut? Yang berikutnya saya ingin bertanya tentang bagaimana kita bisa mengidentifikasi biji berdasarkan ukurannya yang kemungkinan ini dia dari family rumput-rumputan gitu. Terima kasih.
Sorry. So she's wanna ask you about flotation. So because sometimes when you do flotation, some sample are not burnt or half burnt. So is it the unburnt or half burnt samples could be considered as an intrusion or like maybe new
Okay.
samples?
Yeah, this is a really interesting question. A good question. It depends on what period of time you're looking at and the context that your sample has preserved in. For instance, in the Pacific, I've worked with my supervisor, PhD supervisor, Andy Fairbairn, on samples from, the Bismarck Archipelago and further out, where you get some dried, some burnt, some half. And it looks like that is all coming from human behavior, and we're just seeing the spectrum of it, whereas if the site was older or in a less well-preserved site, you would only see the charred part of it. So you have to think carefully about what bits of material you're getting, because little rootlets you'd expect to come into the site just because there's plants growing on top of it. Or, you might get leaves and flowers and things falling in while you do flotation. So there's some things we can discount, but sometimes these are definitely part of that archaeological assemblage. And one of the best ways to work with that is to get dates on them, and then you can be sure.
Perlu ditranslate? Nggak perlu ya kayaknya. Hah? Oke ya. How you differentiate the new or the younger sample, younger seeds and the old seeds or the...
Again, this is all about archaeological context. So, in a site, we have the layers, and this one should be older than that one. Not always perfect, right? And so you can see this with other ways that you look at the site. Is there a lot of bioturbation from animals, from people digging burials? Should we be looking for problems? And if so, then directly dating is 100% the way to do that. Or, like we could look at things like if there is an OSL, optically simulated luminescence sample, in the same, near the context that you're looking at the archaeobotany. If there's no movement of sand grains, if you get a really nice OSL, then it should be, the archaeobotany should be in context because they're bigger than the sand grains, so they shouldn't. So there's a whole lot of different ways that we can think about it. But like anything else, it's about archaeological context and working out if it's in situ or not. And that would be the same with lithic artifacts or with pottery. They can all move through the site. So we have to be careful.
And last question, I think, yeah. How you differentiate between the seed based on its size? Is it possible, or?
The seed based on its size.
Like specimen, probably, like species.
So I think it is based on a mixture of things that you would differentiate different seeds. You'd work with a modern reference collection, either one that exists or one you've made. And it would be the shape, the texture, you can also look at seed anatomy and look at where the endosperm is, and that can give you a good idea of which family it comes from, because that is part of the reproduction, and so it fits very nice with taxonomic lineages. And then size is important as well. And so, you know, for instance, differentiating between a wild Echinocloa millet and a domestic Echinocloa millet is mostly about size. So yes, it's one of many lines of evidence we use to identify. Yes.
Ada pertanyaan lagi? Mungkin? Enggak.
Thank you. Amarlia from Sanggaran Early Museum. Thank you, Dr. Stefani, for your presentation. It's very interesting. And I have a question. Do you find any extinct species of plants you have already found along the time period? It's so long, and we know that Southeast Asia have a high biodiversity, and you just not find the kind of species of plants, but you also find how the people process the plants. And in your cave, if I'm not wrong, you said that you found any process of toxic plant, and maybe you can explain the details.
Oh yeah, so okay,
Thank
so
you.
first question, have I found extinct plants? Not that I know of. At Maja Baby, in the oldest layer, we find a plant that doesn't grow near the site today, but it grows somewhere else. But it's just that vegetation community doesn't exist near the site anymore. But it's still present, just sort of like locally extinct, I guess. Not that I know of is the answer. It's possible, and I think the further back in time you go, and especially local extinctions where you have an environment change a lot because of the economy changing and the clearance of rainforests and those sorts of things, it'd be very likely to find local extinctions, I think, in the archaeology. At Nia Cave, that is not my work. That is the work of Victor Paz, who's in the Philippines, and Hugh Barton, who was, I think, who's now at Leicester in the UK. And they worked together for their PhDs on that site. They use both microfossils and macrofossils. And I think some of their identifications they've pushed too far, but they have this lovely material of Pangium edule. And actually, it's something I'd say is I'd really love if they published more photos so that we can see it and we can see why they made the identifications. But that is my own little wish. But they say they have Pangium edule, and they have it in a context where there is a pit. And we know from the ethnobotany, so from the sort of literature that people were talking about, that Pangium edule can be processed in a number of ways. And one of those is by leaving it to sort of dry, ferment in like a pit, a clay pit. So it's still leaching, but in soil. And that's done over 40 days. And they suggested that finding the nutshell in a pit, like, to process it that way. Is that also suggested that they had a type of toxic yam, but I'm not fully comfortable with that identification, because I think that yams are very hard to ID from each other, and I'd like more information to see how they did that. But there's potentially 2 toxic plants at that site.
Maybe, do you have any idea from these findings, maybe from archaeobotany, and then you go to the biomolecular analysis, maybe to...
It's a really interesting idea. We did a workshop at ANU recently about biomolecular analyses on pottery. And there's been some really beautiful work where people have looked at the charred food crust on pottery, both with archaeobotany, like a scanning electron microscope, anatomical analysis to see if they can see any of the constituents in that food crust, any of the bits of plants that make that up, alongside lipid analysis and isotope analysis on the lipids. So yes, you can do those together in concert. And you could, technically, I think you could potentially do that on a piece of plant macrofossil, but I haven't seen it done before, but it's an interesting idea to do lipids or proteomics on something like that.
Thank you.
Monggo, Pak.
Terima kasih Pak Anton. Thank you very much for your presentations. I just think about the migration waves in Southeast Asia. This area was migrated in 4 times, 4 migration waves. Began with the migration of Homo erectus from Africa to the island since 1.8 million years ago. And then the coming of the anatomical human, modern human, yeah. Then the last one is, I think, Austronesians, coming from the Taiwan until Madagascar, Polynesia, and also New Zealand. Yeah. Your study is very important here, because Southeast Asia consists of mainland of Southeast Asia, for example, in Thailand, in Vietnam, and also island Southeast Asia in Indonesia. And Indonesia, we have all 4 migration waves, since Homo erectus, the oldest one, until the Australasia. And what I think is, how can you difference based on the alimentations, food, traces in the fossils, for example? 2, 1, this is coming from mainland Southeast Asia or in island Southeast Asia. And the second one is, you talk about the Australasia, but how about the earliest migrations? I think Homo erectus, another human, and also Australomelanesis, for example. Thank you very much.
Okay, so thank you for the question. So yes, I talked about Austronesians, but also I think that plant foods are very, very important for understanding the differences between those 2 early migrations. I think, for instance, I suggested in my talk that maybe one of the things that makes humans more able to keep moving into new islands across Wallacea and into Sahol is not just the ability to have maritime technology in boats, and to specialize in sea resources like fish and shellfish, but that is also the ability to process plant foods, because we have a lot of good evidence for people intensively using and processing plant foods straight away in Australia. And we have some evidence in Highland New Guinea and in Naya Cave in Borneo that also sort of connect with that, that corroborate that evidence, although we have nothing yet from island Southeast Asia. We hope to have some soon. But because we know that humans cannot subsist on lean protein alone. And especially in those islands in Wallacea, like Timor and Alor, and especially the very small ones at the end of the Lesser Sunda Arc, where there is not a lot of terrestrial animals, and we know people were using a lot of marine foods that are very, very lean proteins, so shellfish and fish. I just think plants have to be a very important part of that story. And there is some really interesting and tantalizing evidence from Liang Bua, from Thomas and Mike Morley, which suggests that Homo floresiensis there did not have fire and and Homo sapiens, the earliest Homo sapiens, had fire and had lots of fireplaces. And so what does that mean for processing foods? Well, it's important for animals as well as for plants, but it suggests that by the time we have Homo sapiens moving through this area, there is a shift in the abilities to process, to use fire for a whole range of reasons, but one of them is to process foods and to cook. And that changes the sorts of nutrients you can get out of plants. It changes the bioavailability. And so I think that is really important. And the more that we look at plants, I think we're going to find they are a very important part in the ability to process them and to get things like, to get the full use of carbohydrates and fats from plants, whether that be by cooking them, by grinding them, by just opening hard to open plants and extracting things from them, or very, very complex processing sequences like pangeomedule, which requires leaching. I think the more we look, we're going to find that's a really important part of the story of early modern humans in this part of the world, and maybe a bit of a difference with Homo erectus. But I'm sure Homo erectus used a lot of plants. I just wonder if the difference is the ability to process them.
Yes, the oldest culture is hunting and gathering. And the last culture is agriculture. Yeah. But when I see all the